In this set of lectures, we're going to review the cellular level of organization. So we just finished the chemical level. Now we're on to the cellular level. After the cellular level, we move on to the tissue level of organization. If you've had a biology course in the past, either in high school or at the college level, many of these things we're going to learn about in this set of lectures you've heard of. We're going to cover cells and their organelles, so organelles being structures within the cell that perform certain tasks. We're going to learn about the plasma membrane, the boundary of a cell, and transport mechanisms, mechanisms by which molecules enter or exit the cell. And then finally, we're going to review a little bit about mitosis and meiosis, which is how cells replicate to form more cells, or how cells produce gametes, sperm and egg. The difference between this set of lectures and your biology course is I'm honing in on the most important aspects of cell biology for this course in anatomy and physiology. So I will be pointing out why it is important to refresh yourself on these topics. What I like to use is this table. This table can be found in your book. And we're going to go through this table and just briefly point out some of the major parts of a cell. First one being the plasma membrane. If you recall, the plasma membrane is made up of phospholipids. It's a phospholipid bilayer. Inside the fatty acids, you will find cholesterol. So if you think about it, cholesterol is a lipid. Fatty acids are lipids. Lipids typically like to be with other lipids. So the cholesterol will be found within the fatty acids of the phospholipid bilayer. There might also be some glycolipids. So a glycolipid is a lipid with a sugar group attached to it. And it surrounds the cytoplasm. So the cytoplasm being everything on the inside of the cell. Function of the plasma membrane is to basically separate the inside from the outside of a cell. It needs to allow certain things to enter and exit, so those are called channels or transporters. So we're going to be learning a little bit about those guys. There might be receptors on the outside of the cell, which is how the cell might communicate with the outer environment. There are some enzymes in the membrane. There are cell markers we'll talk a little more about. But its overall goal is to determine what enters and exits the cell. Cytoplasm is everything inside. So cellular contents between the plasma membrane and the nucleus. So it's everything except the nucleus and the plasma membrane, to keep it simple. We often call this the intracellular space or intracellular fluid. And it's going to contain a lot of different organelles that we're going to learn about. What are some of these organelles that are going to be important for anatomy and physiology? Now, there are a lot of organelles that you've possibly learned about, but it's not a huge emphasis in A&P. But I'll remind you that there are structures called centrosomes. These centrosomes are involved in the process of mitosis, how cells divide. Cells might have cilia or flagella, which allow them to move. All cells have to have ribosomes. That one has an asterisk because production of protein is extremely important for the cell. So ribosomes are these structures that read the RNA and determine what the sequence of amino acids need to be for that particular protein. In the world of microbiology, you'll learn that certain antibiotics target the ribosome in bacteria, kind of inhibit the ribosomes of the bacterium, doesn't affect our ribosomes. Therefore, these antibiotics are very specific at destroying bacteria. The ER, you've probably heard of the ER. The ER is kind of made up of these membranous, flat organelles that surround the nucleus, and there's two types. There's a smooth and a rough ER. The rough ER is involved in producing proteins, whereas the smooth ER is more important in producing fatty acids and steroids. When we get into the liver, we're going to learn that the liver has a very, very robust ER. A lot of ER in cells of the liver. And that's because one major function of the ER, the smooth ER, is to detoxify. And your liver detoxifies your blood. What else? The Golgi complex. You've probably heard of that. The Golgi complex is often thought of as kind of like going to FedEx and giving them a package and they help you send it out. Again, a lot of these membranous sacs, and it takes in proteins from the ER, kind of modifies them, and then ships them out to their destination. The lysosome, extremely important when we get to the immune system. So when we get to the immune system, we're going to learn that we have certain types of cells called phagocytes that can go gobble up bacteria and destroy the bacterium. And one way to destroy that bacterium is to use a lysosome. A lysosome has a lot of enzymes that can destroy and break down things. So you can use the lysosome to help destroy the bacterium. Not going to talk too much about a peroxisome, a proteasome, but mitochondrion is a very important organelle. And we've probably heard of the phrase the powerhouse of the cell, and that is because the mitochondria is the site of aerobic cellular respiration. So cellular respiration is production of ATP. Aerobic means it requires oxygen. So all that oxygen you breathe is trying to get to that mitochondria. And that oxygen helps facilitate the production of ATP. Finally, this slide just mentions the nucleus. I think we know the nucleus contains your chromosomes, of which a human cell has 46 chromosomes, or 2 sets of 23. Things have to be able to enter and exit that nucleus, so there are little pores in the membrane that forms the nucleus. But that's where you're going to find your chromosomes that contain your genes. Next topic is going to be the plasma membrane. We'll cover the plasma membrane and how molecules enter and exit the cell in the next video.